Hello, we're at section 1.3, and we're going to talk about the process of a statistical study. Now, right off, I'll tell you this might be a little bit on the long side. The good part is I've added some graphics, maybe even some cartoons, so hope I can keep your interest here. It is very important because even if you're just reviewing a statistical study, you know some of these things just to make sure that it's been done correctly. And yes, go ahead, call me crazy, but actually I enjoyed reading this section in the book. So give it a shot. In fact, I would suggest that even if you read through it real quickly, you read every section in the book as we go through it, not the ones we don't cover. Here's a little picture. We're going to talk about the process and various kinds of studies. At the top right, I've put a sort of a letter summary to help you maybe remember. Unfortunately, DCOA doesn't represent a word, but it does represent the order in which you're going to conduct this statistical study. Here's another graphic showing you starting a study design. Decide whether you're going to do an experimental study or an observational and so on. A good picture. We're going to cover several of these things, not all of them. So, first question. We want to study the effect of vitamin C on nerve disorders, and we'll do several things with this particular example. So keep this in mind. There's lots of ways of getting vitamin C. We're going to give an IV dose, intravenous dose of vitamin C, and see what happens to nerve pain of the patients. This will be limited to patients that only have specific types of disorders. What were we asking for? I'll go back this time. It says identify the population of interest and the variables. Population is the patients that have those disorders, and the variable is the amount of vitamin C and the amount of nerve pain. Two kinds of studies. You need to keep these in mind as you're reading studies. An observational study, you just look at the data that are already there. You don't actually make any changes yourself. With an experiment, you generate data by identifying cause-effect relationships by actually doing something with your subjects. So a quick check to see whether you make sense of this. See which kind of study each one of these is. The average age of college students across the nation, would that be an observational study or an experiment? Take a sheet of paper. You'll need this as you go through this and most of the lessons. Write down your answers. That way you actually know what your response was. You know whether you get it right or wrong, and you don't tell yourself, oh, well, I meant that, and think you got it right. Write it down, please. Okay? So what do you have for these two? Well, A, it's an observational study because you can't change the ages. All you're doing is collecting data that already exists. Second part, it's going to be an experiment because you see what happens with flu shots and flu prevention. A representative sample, extremely important. You want your sample to look like the population, but in a small number of them, a subset of them. Make sure that your sample doesn't favor one group from the population over another. For example, take a look at this. On the left-hand side, see get a pen here. On the left-hand side, we're talking about this part. Notice that on the left-hand side, this sample, or this population rather, is about 50-50 pink to blue. The sample isn't 50-50, so it's not representative. On the bottom part, bottom right part, notice that the population here is very similar to the population here. Most of those are reds, female, with a few males. And down here in the sample, you have mostly reds with a few male. Here's another example. You want to know how 18 to 25-year-old women spend their money. I'd like to know that too, but fortunately, I don't have any daughters that are 18 to 25, and my wife isn't 18 to 25 either. I'll tell you that right off the bat. So which of these groups would you use as a sample? Would be A, B, C, D. On your piece of paper, choose one of those four. Pause if you need to think about it, and then move on. You should have chosen A, 18 to 25-year-old women at the mall. Part B is men and women. We don't care about the men in this particular study. C, women of all ages? No, well, we don't want all ages. We only want 18 to 25. Part B, why didn't we choose that? Well, it's women commuting to work, not all women, 18 to 25, so that was out. So A was a good answer. Hope you got it. Now, sampling techniques. We've got 6 different sampling techniques. They are different. The first 2 are easy to mix up, so be careful. Number 3 and number 4, the stratified and cluster, are easy to get confused between the two, so you have to pay special attention to those pairs of types of samples. The last 4 are pretty easy, systematic and convenience. We'll get to that now. Okay, the random sample, the first one. Remember, I said the first and the second one are easy to mix up if you're not careful. So be careful. For the random sample, this is what you might expect. Every member has an equal chance of being selected. So we have a picture of a bingo being played, and any of those numbers can be chosen at any particular pool of those tokens or whatever you call them. 16 can come out, 72, 53, any of those have the same chance of being selected. It makes it a random sample. Now, notice the difference with this one. In this one, you're picking up one item, and any one item has the same chance as any other one item. Next, simple random sample. Simple random sample. So notice this is the difference between these two, and what does that mean for us? It says every sample has an equal chance of being selected. So what does that mean? Well, look at this group of smiley faces or faces here. We've got 4 by 4, we've got 16 different faces. Now suppose we want to pick 3 faces from that population of 16 faces. There are a lot of different ways we could pick 3 out of the 16. A simple random sample means that there are a lot of different ways we can choose them, but every way we can choose 3 out of the 16 has the same chance as any other 3 out of the 16. This might take some extra thought. It's not quite as simple with a simple random sample. Next, stratified sample. I already said that this one and the next one are easily confused if you're not careful. So pay particular attention and create your own example so that it's easier to remember. So the population for the stratified, think of this stratified. If you're at the Grand Canyon and you're looking at one of the walls, you can see different strata, different colors. Well, in this small example here, we're talking about the strata being freshmen, sophomores, juniors, and seniors. What we want to do is split this group of students up into those 4 groups. And within each group, we pick the same number of subjects. So we're going to get 2 freshmen, 2 sophomores, 2 juniors, and 2 seniors. This way, when we look at our sample, we've got some of each and the same number of each. We don't accidentally get 1 freshman and all the rest seniors, or some bad mixture like that. So we want everyone to be represented. Now, here's the other variety. We just talked about the stratified. This is the cluster sample. On the other one, we start out by grouping them, so every group gets selected. In this one, we look at different groups, sort of like zip codes, for example. Suppose this is rice in zip code 1, rice in zip code 2, rice in zip code 3, and so on, or plots of ground, whatever. Now this is the important part, every one of these plots looks like any of the rest. If someone put you down into this plot, there you are, you're in the plot. If you just look at the plants, the rice plants around you, it looks the same as if you were over here instead. All those plots look the same. Since they all look the same, what you'll do is you'll pick whole clusters at random. So in this case, we'll pick this field, this field, this field, and this field, those 4 fields. We pick those at random. We don't worry about the rest because all the rest are the same as the ones we picked. And we use that collection or that set as our sample. Got that? All of these individual sections look like all the rest. So if you're in this field, it looks exactly the same as in this field. Now we get to the easy one, systematic sample. Select every nth member of the population. So in this case, we're going to sample some bottles of soda off the assembly line. We pick every 4th bottle. That'll be our systematic sample. It can be every 4th one, could be every 100th, or whatever we choose. And the last one that we're going to cover, because there are many different ways of sampling, but these are some of the more frequent ones used, a convenience sample. You might call this the easy sample. Now there's some good reasons to do that. You can get the data quickly, it's cheap, It's easy to do the research, cheap again, readily available. Some of these are the same as others, and fewer rules. Here's an example over here on the right. This is a group of people. Imagine that these people are, let's say, at a football game. And this is you. It's a young lady here, or a gentleman with long hair. And you decide, okay, Mr. Sandler said I need to get a sample of 5. So I'll just get the 5 people nearest in the stadium. And that's my sample. That's a convenient sample. Taking it easy, but not being careful as to whether that sample is representative. The difficulty arises when the ones you choose don't look like the big group as a whole of the population. Your sample should look like the entire population. If it doesn't, it's not representative, but it is convenient. I'll give it that. Hit the pause button. You can read through this yourself. I'm not going to annoy you by reading it for you. And then continue when you're ready. So, some quick examples. If you need to, slide back, make a list of those 6 different kinds of samples so you can pick which one you think is which. You're going to put your answer to the right, something like that, or just write down A, what kind of sample, B, what kind of sampling method, C, what kind of sampling method, and so on. There's actually 5 of them. Excuse me. You got that? Pause if you need to. There's 2 more. Get those at the same time. Categorize those with the 6 different types that we're talking about. Pause it until you have all 5. And here are the answers. First of all, first one is the voting precincts are the strata. Second, every 10th box, systematic. That's an easy one to pick out, usually. Convenience. That didn't take much effort, did it? D, cluster sampling, because you're going by school districts, and every school district you're assuming is like every other school district. Random sampling, every name in the box has an equal chance of being chosen. Notice the one we didn't have as an example was one of simple random sample, where every possible subset or sample has the same chance of being selected. Next, 2 different kinds of studies, a cross-sectional versus longitudinal. I don't have any quick tricks for you on this one, but the important thing is with a cross-sectional study, you're talking about 1 point in time. So stop your stopwatch or stop your watch at 2 o'clock today and take data at that particular time. For a longitudinal study, you're going to look at maybe 1 class over a period of time, so maybe over the whole semester. It would be a longitudinal study. The cross-sectional study would be studying maybe this class on the first day of class or the 7th day of class. Longitudinal would be studying the class over the whole semester. So let's see if you got that. Have your paper and pencil ready? Which is it? Cross-sectional or longitudinal? As soon as you're ready, move the video along. The first one, they followed them for a period of time, so longitudinal study. Hey, I've got something for you here. maybe this will help. The L in longitudinal and the T in time. If you're not careful, these two can look very similar, right? Except for the crossbar. Maybe that'll help. I don't know. It's up to you. Second part. Snapshot means it's a cross-sectional study because it's a specific point in time. So you stop the clocks and you took your study at that particular time. Next, both of these are observational studies. Now, we're going to get back to this, but you have observational studies and you have another type, and we'll get to in a moment. But for the observational studies, now this is fairly simple. The case study looks at a single event. The meta-analysis uses studies that have already been done. Here's a graphic for you. This is a meta-analysis because we're combining the results of individual studies. Here's study number 1, here's study number 2. We're going to take those and put them together into our meta-analysis. Case study. You're going to follow them. You're going to do one study. It's a case study. Which one is which? Where the case study is one. The meta-analysis means you've got a bunch of case studies put together to make one big study. You got your answers written down? You ready? First one, they're looking at multiple studies, so it's a meta-analysis. And the other one, the case study, because they're talking about a single tsunami. Next. Now the treatment is something we're doing. Now remember, we just talked about observational studies. This is an experimental study, because you're applying a treatment to some subjects. For example, suppose you have a new type of aspirin or pain reliever, and you give that pain reliever to some people that have a particular condition that's painful, and we're going to see whether it helps their pain. The treatment would be the actual new pain reliever, and the subjects are the participants in the experiment. Now the data didn't already exist, so it's not an observational study, it's an experimental study. Subjects, obviously, are the people or things, could be 2x4s, and you're painting with something, seeing how long the paint lasts. Participants, pretty much the same thing as the subjects, except they are people, they aren't things. Now, if you looked at the original slides, these last two are in a different order. I put them this way because the explanatory variable is sort of what you think of as coming first. So, for example, in the case of the pain reliever, the actual aspirin or ibuprofen or whatever it is what you are using. You give it to the people, and the response variable is how much pain there is after you've taken that pain reliever. Explanatory is the one that comes first. Response is the one that comes second. So here's a little bit of a picture. I've got lots of extra pictures from the originals. So hopefully this helps a little bit. The experiment imposes some treatment on individuals. It doesn't have to be people, remember. It could be objects of some kind. So you have a subject, you perform a treatment on them, and then see what happens. Here's some, you can pause here and read through these. This is another way of giving you an idea of what the explanatory and response variable is. Explanatory variable, response variable. So you're imagining usually that by doing whatever this treatment is, you're going to create this result. Sometimes they're connected. Well, we'll see. Get back to that in a moment. Principles of experimental design. Now, on the left here, notice I put the 3 letters. Randomize the control and treatment groups. Control outside effects. In other words, we would like to have just one effect for a response variable. One response variable creating the effect. Also, you want to replicate it. In other words, do the experiment to see if you get the same result or meaningful patterns each time. Otherwise, you might have to go back and redesign your whole experiment. So, here are some things you need to remember. The control group is a group that's in the experiment, but no treatment is applied to those subjects. Control, no treatment. Control, no treatment. Sorry about the letter there. You can fix it up in your notes. The treatment group is the ones that actually get the treatment. In our pain reliever example, those people get the actual medicine that's supposed to relieve the pain. The ones in the control group get something that might look like it, but is actually not the actual treatment itself. Now this last one, confounding variable, can be confounding itself if you're not careful. Those are extraneous factors that mess up the connection between the explanatory and the response variables. We've got an example or two for you to see. First of all, we've got an experimental group and a control group. In other words, this group is having the actual treatment done. The control group does not have it done. But the important thing is, it looks like the treatment is being done to both. Now I've adjusted this a little bit. Notice that in this first one it says biofertilizer X is sprayed. You've got these little dots here. That represents maybe the chemicals that are included with that spray. So you spray it on, see what happens to the plants. Do they get better, or maybe you're trying to get rid of pests. Let's assume you're trying to make them grow bigger. See what happens with this group. Over here, these strains here represent just water. No chemicals at all, and see what happens to the growth of those two plants. Does it do better with biofertilizer X or without biofertilizer X? Now, let's think of the control group and intervention group as Let's suppose we've got a headache drug. I'm going to call it Ache Away. I guess sort of funny. I didn't make that up though. I'm not responsible, except that I did put the slide in here. Okay, so the control group, what happens? They're going to take tablets, and the intervention group is also going to be taking a tablet. If you're just watching them, you don't know which one is taking the actual medicine and which one is taking a fake. The placebo group, or the control group, is taking a tablet every night, but it might be a salt pill or a sugar pill, or something like that. Or maybe just, whatever, you can make it up yourself. Notice that 5% of those people, even though they're not really taking the medicine, 5% actually feel better. They think they're taking the medicine. Over here with the Ache Away tablets, 85% feel better. It sounds like Ache Away might be pretty good. You want to go out and get some this afternoon? Anyway, the results are the Ache Away is more effective than a placebo. Placebo made 5% feel better. The Ache Away, 85%. Let's go get some of that. Before we leave this, we're going to get back to it too. But this is the placebo effect. You're taking fake medicine, is what I would call it, but they get better regardless of whether there's actually medicine in it or not, because their minds say, oh, I'm taking the medicine, and they feel better. Moving on. Confounding variables. So what are they? Well, first of all, let's look at the bottom. If we just look at ice cream consumption and the number of sunburns, notice that when there's more ice cream bought, there are also more sunburns. Now, if you just look at those two, you think, oh my gosh, the more people eat ice cream, more sunburns they get. However, there's a confounding variable. The confounding variable is the hot temperatures. The hot temperatures are affecting the ice cream's consumption and the sunburns. Higher temperatures, more people buy ice cream. Hot temperatures, more people get sunburns. The hot temperatures are affecting both. It's not the ice cream consumption causing the sunburns to go up. And if for some reason you think of it in the opposite direction, which you might say, well, I've got it. The more sunburns you get, the more people eat ice cream because of the sunburns. That's probably not true either. The hot temperatures are the confounding variables. Or the variables, sorry. This is a general look at it. Like what you might do is you might take that slide and make up your own example. Next, we mentioned this. The placebo appears identical to the treatment. It has no value whatsoever. The placebo effect is the result of the power of suggestion, because you're not getting the pain reliever, but you feel better anyway, because you thought you were taking it. And, excuse me, a single-blind experiment, double-blind experiment. Well, one good thing about keeping these straight is in the single-blind experiment, you're talking about one thing. The subjects do not know which group they're in, the control group or the treatment group. The subjects don't know. It's one blind. In the double blind, the subjects don't know, and the people interacting with the subjects, the nurses, for example, they're handing out the medicine or non-medicine. They don't know whether they're giving the pain reliever or the fake pain reliever. Single blind, the subjects don't know. The ones administering the treatments do know. Double blind, the subjects don't know, and the nurses don't know, let's say. So here's a little bit of a long one. You might read through this, you might have to go back, because there's 2 pages on this one. This is talking about the vitamin C again that we talked about at the beginning. You might hit pause here and then move on to the next one. Now, in Group A, they're getting the IV doses of vitamin C. And we're going to track what happens to the nerve pain. Does it get better? Nothing? Does it get worse? Group B, they get IVs of saline. Well, that doesn't have any ingredients at all that are going to have any effect on the patients. We're going to find the amount of pain reported by the separate groups, the treatment group, and control group. If necessary, go back and answer these 5 questions. What's the explanatory variable and the response variable? What is the treatment? Which group is the treatment group? I think I might have given that away. Sorry. But do you remember? Or do you agree? Could I have made a mistake? What's the purpose of administering saline to the group B? And is it a single blind or a double blind experiment? Study. And do you think that's the best choice for that study? So, let's take a look. Explanatory is what explains the changes in the response. The explanatory variable is the dose of vitamin C. Response variable is how much nerve pain there is. Get a circle around that. Amount of nerve pain. That's the response. There we go. The treatment is the dose of vitamin C. A is a treatment group because they actually got the vitamin C. B is a control group, they just got the saline. Saline is a placebo, and that's to suggest to those people in that group that they are getting the treatment, the vitamin C, when in reality they're not. Maybe if they're lucky, they'll have a placebo effect and there will be a positive response. Don't expect it though. And last of all, it's a single blind study because those administering it knew which of those shots they were giving, the saline versus the vitamin C. Last of all, very important too, informed consent. And you sign the papers saying that you want to participate in the study. You don't study someone without their agreement. And the Institutional Review Board is what looks at how it's been studied and see whether it's appropriate and make sure that no harm is done to the subjects. Well, we've finally got to the end, and we'll see you next time for section 1.4. Bye-bye.